This is our last module for the NAS to review, and in this module we will cover inheritance. Humans have 23 pairs or 46 total chromosomes. Chromosomes are made of DNA, deoxyribonucleic acid. A karyotype is just a picture of the chromosomes, and they're arranged in order in pairs from 1 to 22. Those are called the autosomes, 22 pairs of autosomes, and they code for physical characteristics like eye color, hair color, whether you have dimples or don't have dimples. And then you have the 23rd pair, which is either going to be XX or XY, and those are going to determine sex-linked traits. A gene is just a segment of DNA that codes for a specific protein, and then an allele is a copy or a variant of a gene. So for instance, in our chromosomes, we get one chromosome from our mother and one from the father, and that makes up the pair. Let's say that pair, within that pair, there's a gene that codes for dimples. Now, the allele being a variation of that gene, let's say one chromosome has the allele that codes for dimples and the other has the allele that does not code for the protein that is eventually going to result in dimples. So the allele is just a variant of the gene. Genotype is the exact genetic makeup, what the alleles are, and then the phenotype is what you actually see. Here's a karyotype, and as I said, they're arranged in order. Number 1 is the largest, and you can see that these are in pairs. We call these homologous pairs. One of those chromosomes is maternal from the mother, the other is paternal from the father. The last two chromosomes down here that you see are X and Y, so this would be a male. Again, allele is a variation of a gene, so a gene codes for a particular protein. You can have different alleles for that gene. And in the example here, we have for eye color, and again, you can tell which chromosomes go with which by looking at their size. So this is a homologous pair of chromosomes. This one has the allele for eye color being blue. This one has it for brown. Two different alleles for that gene. Mendelian inheritance is a strict dominant recessive inheritance, meaning that one allele, the alleles can be dominant or they can be recessive. And one allele is going to be dominant to the other. If a recessive allele is going to be expressed in the person, both of those alleles must be recessive. Otherwise, the dominant is always going to be expressed. The way we write these alleles down, the dominant is in uppercase, recessive is in lowercase. For instance, dimples. Dimples is a dominant trait, so I'm going to use the letter D to represent dimples, and the dominant allele is going to be uppercase D. Remember, you have two alleles, so a person could have uppercase D, uppercase D. This is what is called the genotype because it's what the genes show. We would call this homozygous dominant. Same allele on both chromosomes. The phenotype is what gets expressed, and it's going to be dimples. Okay. Here I have another person, and in this case, one allele is the dominant, the other is the recessive. We would call this heterozygous, hetero meaning different. The phenotype here is going to be dimples because the dominant allele gets expressed in this Mendelian inheritance. Third person, two recessive alleles, two lowercase. This is homozygous recessive. They're the same, homo meaning same. The phenotype here is going to be no dimples because there's no dominant allele and dimples is the dominant trait. So just keep in mind the difference between genotype and phenotype. Genotype is exactly what the alleles show, whereas phenotype is what you can see, what the trait that you see. Now, some disorders are autosomal dominant. For instance, Huntington's disease is autosomal dominant, meaning if you have the allele for that abnormal gene, you will have the disorder. Some disorders are autosomal recessive, so the only way to have that disorder is to inherit both recessive genes or alleles. And sickle cell disease is an example of that. I just want to show you Punnett squares. A Punnett square is a way of uh, looking at how two parents with their different alleles and the possibilities of what the offspring might have. In a typical Punnett square, you will put the maternal alleles on the top, the paternal on the side. And you can see these are paternal because they put the little flagellum here for a sperm. And remember that each person has two different alleles for whatever gene it is. There's two different alleles. So this person has uppercase B, lowercase b, and I don't know what the trait is. It could be anything. And then on the father's side, it's also uppercase, lowercase. And the way you do Punnett squares is you bring one down and one over, and so this individual would have both dominant alleles, homozygous dominant, okay? And whatever that trait is, if it's a Mendelian inheritance, they would have that trait. This possibility, you can bring this down, you bring this one over, and you have a heterozygous. And the same thing for this, also heterozygous, because you're bringing now this one down, this one over. And again, in Mendelian inheritance, dominant trait is there. If the dominant trait is there, it will be expressed. So these two individuals would express the dominant trait. The fourth square here, again, bring this one down, this one over, and you notice that this is a homozygous recessive. So whatever that trait was, dominant trait was, it's not going to be expressed. Whatever the recessive trait is, it will be expressed. If you want to start looking at percentages, you can do that. And if you want to look at, well, what's the chance for any particular pairing of these two parents for that one child to express the recessive phenotype, it would be 1 in 4. Okay, 1 in 4 right here. Let's go over to this one, and they've changed the letters, but you can see that the mother is still heterozygous for the trait, and the father is heterozygous. So they're both heterozygous. What is the chance that any one particular offspring will show the dominant phenotype? Well, it's 3 in 4, or 3/4 of a chance, 75%. Because if you do the pairings here, you see that 3 out of 4 times you're going to get the dominant allele here. And whenever you have the dominant allele in Mendelian inheritance, it will be expressed. How about non-Mendelian inheritance patterns? And some of these are the sex-linked traits. They can be X-linked, meaning that the gene will be on the X chromosome, and that can be dominant. You can also have X-linked recessive, or you can have Y-linked. Y-linked is pretty rare. X-linked dominant is fairly rare, but it does happen. If the trait is on the X gene and it's dominant, well, it doesn't matter if it's a male or female, it will be expressed because it's the dominant trait. For X-linked recessive, it's a little bit different. If a female has just one of those alleles, one of those recessive alleles, they're considered a carrier. Okay, they can pass that down to a child, but they aren't fully effective with whatever that inheritance is. They might be a little bit, but not fully. And the reason is they have a normal X chromosome. So if you have a normal X chromosome, you can make up for the defect in the abnormal one. However, for males, if they have that X-linked recessive gene or allele, they have no other X chromosome to make up for it. They just have a Y. Okay, so they will express that trait. Easiest way to look at this is color blindness. Color blindness is an X-linked recessive trait. You'll notice this Punnett square looks a little bit different than what we had before. That's because the X-linked traits, the letter is put as a superscript. Okay, so the female is still on top of the square here, and the male is on the side. The letter we're going to use is a B. So there's a uppercase B, lowercase b. Lowercase b is color blindness. So we notice here that the female is heterozygous for it. This female would be considered a carrier. Now the male has the dominant allele on his X chromosome. He has no other X, so there's no other allele here. This father is not colorblind. He has the dominant allele. So if we do the pairings, again, you go down and over and you can get 4 possibilities here. Well, first you can notice here that for any one child, there's a 50-50 chance that it's male or female. But the other thing you notice here is there is a, for the females, okay, for the females, there's a 50% or 1/2 chance that there's a female carrier, and that would be this person right here. Now for the males, there's a 50% chance that a male will be colorblind. Again, he has this recessive allele, but there's no other allele on this Y chromosome to make up for it. And the difference between the male here and the female is that the female has another X chromosome with the dominant allele that can make up for the abnormality. Codominance. When we talk about blood groups, on our red blood cells, or erythrocytes, we have different things sticking out of them that are called markers, and these are genetically determined. These markers let our bodies know what is self and what is not self. Okay, so if we look down here at the bottom, a person with type A blood is going to have this A marker on it. A person with B is going to have a B marker on it. A person who is type AB has both. This is codominance, okay? A and B are codominant, meaning that if you have an A allele and you have a B allele, both will be expressed. And that's how you get a type AB blood. Now, A and B are dominant to O. So a person with type O blood has neither A or B. And I know this is confusing. We'll go into it more when we get to that chapter. But just keep in mind that for some traits, there's a codominance where both get expressed. Now, as an aside, if a person has type O blood, then both of their alleles have to be O. It's a recessive. Genetic variation. I mentioned in a previous module about crossing over in meiosis. This occurs early in meiosis, and one chromosome can exchange genetic material with another chromosome, and we call that crossing over. And this is why a child doesn't look exactly like a parent. It's a sort of a mix between maternal and paternal genes. And when we look at these pairs of chromosomes, we call this a homologous pair here, but we have these chromosomes have already been replicated, or the DNA has been copied. So one comes from father and one comes from mother. When we do the crossing over, what happens is some of the mother's genes are on the father's chromosome and vice versa. So now when these split up and go into either egg or sperm, we get daughter cells that are going to be genetically distinct from each other. Chromosomal disorders. Just quickly, there's lots of different ways that chromosomes can contribute to disorders. We're just going to talk about non-disjunction. In meiosis, there are places where chromosomes must separate in order to get the correct number of chromosomes in the daughter cells. If you do not have separation of these chromosomal pairs, you can get, for instance, an egg with 24 chromosomes instead of 23. Remember, they're supposed to be 23. Or you can get one with 22 instead of 23. And when that egg or sperm fuses with egg or sperm, you would get either too many chromosomes, 47, in the what we call the zygote or the fetus, eventually the fetus, or you would get perhaps 45. That's too few. Well, let's just talk about trisomy 21. Trisomy 21 is Down syndrome, and it's the most common non-disjunction. In this non-disjunction, there are 3 copies of chromosome 21. Here is non-disjunction. A couple of examples here. It can happen during meiosis 1, but the end result is you get too many chromosomes in a couple of the daughter cells and too few in a couple of the daughter cells. You can also have non-disjunction during the second phase of meiosis. Again, same, you can get daughter cells with too many or too few. And if, let's say for instance, this daughter cell, and we're going to call it an egg, fuses with a sperm that has the correct number, the resulting zygote is going to have 1 too many chromosomes, or 47, 1 too many. And perhaps that extra chromosome is chromosome 21. That would result in trisomy 21 or Down syndrome. Here we have a karyotype for Down syndrome. And you can see here it's circled, there are 3 copies of chromosome 21. And that does it for our last module for NAS 2.